All right, for the benefit of those of us who can't be with us, I'm going to go ahead and record this, and I'll talk you through how I want to do what I think is going to be the last lab, because holy guacamole, it's Wednesday. We got one more day of me flapping my gums at you, and then the exam's on Friday, and then that's all she wrote. And it's all over but the weeping and the wailing. That would be me. That's me, retiring to my study with a very large bottle of alcoholic beverage in your finals. Right. Okay, I have just uploaded this in the usual place. We've already done a lab where I introduced you to cladistics. I always get that wrong. And that was the one where we whispered amusing sentences down a line. And I pointed out that by the time a sentence got to the two ends of our forked line, not only was it very different from what it started from, but the two tips of our branching line sometimes ended up with different versions. And I pointed out that this is just what happens when you transfer information accurately but not perfectly. Copying errors creep in. In genetics, we call those mutations. And I think I mentioned every one of you is estimated to have something like roughly 30 points in your DNA that you inherited neither from your mother or your father. They result from genetic mutations. Most of them do nothing. The vast majority of mutations we now think are selectively neutral. They do create new alleles, but there is no selective advantage or disadvantage in them. Some of those mutations are harmful, of course. Some are, in fact, beneficial. A recent attempt to quantify this, by the way, was done by a guy named Dr. Richard Lenski at Michigan State, who's been growing E. coli in culture for 65,000 odd generations, and basically just watching what they do and how they adapt. And his estimate was that roughly 1 in 12 of the mutations that he observed in his E. coli culture was actually beneficial. There have been several that were observed giving E. coli the ability to metabolize a food source that before the experiment started, they could not use. So there are beneficial mutations, probably more common than we recognize. There are some that are harmful, and probably the great majority don't do anything. And what we can do is use some algorithms to look for signal in molecular sequence data and work out when various mutations happened, and use that as a tool for reconstructing evolutionary history. And the lab handout starts with a little practice data that I've given you. Okay, good. I am sharing the screen. I was temporarily terrified that I might not be. And this is from the handout that I've uploaded in the usual spot. This happens to be a little bit of DNA sequence data from a hippo up on the top, and the corresponding gene off the bat, I think this might be like an NADH dehydrogenase. I think it's a gene involved in respiration. Off the bat, I forget. But it's the corresponding gene in 6 different species of whale. The orca, AKA the killer whale. You know what those things do? Yeah, an orca will swim up to a great white shark and basically just bite it in the belly and suck its liver right out, and then swim off. And you have a shark with a gaping belly wound where its liver got sucked out. Naturally, the shark dies, but dang, I'm impressed. Anyway, hippo, orca, sperm whale. Sperm whales noted for the huge amounts of oil present in a special organ in their head. Sperm whale oil used to be commonly burned for lamp oil up until, I guess, the 1870s would be about when petroleum products started going big time and eventually driving the whaling fleet out of business. But sperm whales, you know, and then blue whales, gray whales, humpback whales, and fin whales, which are all really big. The blue whale is the largest of all mammals. The gray whales are the one that I used to see in the Gulf of California. Humpbacks are the ones I got to see when I went to Boston. If you ever go, the New England Aquarium runs these whale watching cruises. Because there's a spot you can go like maybe 40 miles off the coast, which is a humpback fishing ground or feeding ground. And you can see lots of them. Anyway, so there we have the sequence data. And it's a tiny snippet, but I've given it to you because you can start looking for similarities and differences in that data. So if you look at position 1, you'll notice that the hippo, orca, sperm, blue, gray, humpback, and fin whales all have a phyneme, right? That's a T. Yes? That doesn't really tell you anything. But look at this. Look at the second one. Hippos and orcas have a guanine in position 2. Right there. But sperm whales, blue, gray, humpback, and fin whales have an adenine in position 2. Okay, interesting. Why might that be? Okay. Well, first of all, unless like some of these species are actually aliens from the planet Azorgo, or were separately created by various different gods, then presumably they've all got some common ancestor if you go back far enough. Okie doke, with me there? Why would some of the descendants of that common ancestor have a guanine and some have an adenine? Okay, somewhere in that past history, one of those ancestors underwent a mutation. Presumably not one that had such a bad effect that that ancestor couldn't survive. Probably one that was completely neutral. But that mutation got passed on to all of the descendants. So maybe, just maybe, the common ancestor had a guanine, whatever that common ancestor was. And then the descendant that led to sperm, blue, gray, humpback, and fin whales underwent that mutation and gained an adenine instead of a guanine. Okay, with me there so far? Okay, that seems like pretty big speculation. Can we do any better? Okay, position 3 and 4, everybody's got a C. Everybody's got a cytosine. Okay, position 5 is a little bit more interesting because the hippo has a cytosine, and everybody else, all the whales, have an adenine. Okay, so maybe, just maybe, the common ancestor of all 7 of these species, whatever it was, had a cytosine, and then a descendant of that ancestor on the line leading to whales underwent a mutation and gained an adenine, and that adenine got passed on to all of its descendants. Make sense? Are you following the way the logic breaks? Okay, cool. Okay, here in position 6, hippo's got a guanine. This gets odd because there's a guanine, there are some guanines, and there's a guanine. So 3 of the whales have a guanine. and 4 of the whales have an adenine. That is potentially interesting. Okay, here at position 7, the hippo's got a color do I not get used, has a thymine, and 4 of these whales have a thymine. But the orca and the sperm whale have a cytosine. So maybe in this case, the common ancestor of all 7 had a thymine, and that was retained in all of its descendants except for the common ancestor of orca and sperm, which changed the thymine to cytosine, and so on. Okay, I'm beginning to see signs that you're getting bored as hell. So I'm detecting a kind of dreamy look in people's eyes. So I'm not going to talk you through every last one of these, but here at position 8, everybody's got an adenine. Here at position 9, we have some guanines and some adenines. There at position 10, we've got 1 whale with a thymine and everybody else with a cytosine. Blah, 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 blah. I'm not going to do this for all of the nucleotide positions. Okay. What cladistics does is take a whole bunch of data like this and look for the tree that accommodates as much of this data as possible. Sometimes the data is going to contradict itself. Like for example, here, look at 5. A possible explanation here is that the hippos got cytosine, and that was present in the common ancestor. And then all of the whales had a common ancestor that mutated that cytosine to adenine. This might actually get a little bit clearer if I do this on the whiteboard. Okay, so one possibility is that is that we have a tree looking something like this, and the common ancestor had a cytosine, which I'll draw as a little C right here next to a tick mark, and the hippo still has it. And all of the whales, all 6 of them that I've got, descend from a common ancestor where the cytosine changed to an adenine. That's one possibility. That's consistent with what we see over here in our data, which for some reason has completely lost its colors. But right there, the hippo's got cytosine and everybody else has an adenine in this column. That's not consistent with like the next one, where let's say the hippo's got a hippo, sperm, and gray have a guanine, and 4 of the whales have an adenine. That may not be consistent with all of our data, but cladistics is about looking for the tree that is consistent with the largest amount of data that's possible. Looking for the tree that has the fewest amounts of contradictions in it. You'll always get some. You know, if you think about it, there are only 4 possible nucleotides. And it's quite possible that, you know, if any given T could mutate to a C in completely unrelated lineages. You know, it is quite possible I've got an A at position 5, and I'm not related to whales at all, or at least not closely, and certainly not related to hippos, you can always get lots of convergence. Cladicics is about trying to find the signal in the noise. And it's a colossal pain in the bananas when you are doing it by hand. Fortunately, it's the kind of thing that computers do very well. Okay, are you getting some sense of how the logic behind it works? Okay, you can always find contradictions in the data because that's the way evolution works. You can always get cases where a nucleotide reverts, or where two unrelated species happen to have the same nucleotide change. Just because there's a limited number of nucleotides and so a limited number of types of mutation that can happen. But ultimately, you're looking for sets of organisms that share the most features possible. So the organisms that are most closely related to each other are going to be the ones that are probably will be the ones that share the most unique features. So, for example, I mean, we already know that orca, sperm, blue, gray, humpback, and fin are all whales, and the hippo is not a whale. If that's the case, we would expect to see some nucleotides that are shared by all 6 of these that are not shared by the hippo, because they changed somewhere on that branch line where the branch leading to whales branches off the line leading to hippos. So let's see if we can find them. Go back to, let's go back to draw. Let me see here. Okay, here in position 5, that's a change that all 6 whales share, but the hippo doesn't. Here at position 15, that's a change that all 6 whales share, but the hippo doesn't. Position 12, yeah, that's a change that all 6 whales share, but the hippo doesn't. Do we have any more? We probably do, but I can't. For some reason, it's not letting me scroll over that far. But yeah, we've got 3 changes that are shared by all 6 whales, but they're not shared by the hippo. Now let's see. Here we've got a change that's shared by all taxa except the orca and the hippo. And do we have any more like that? I thought I saw one a minute ago. Here, yeah, we've got another change that's shared by all except the orca and the hippo. Okay, and then here we've got one that's shared by blue, gray, humpback, and fin whales, but none others. And do we have any more like that? For the orca, okay. For reasons I'll get to in a second, that may not tell us as much as we'd like it to. There we go. Of course, unfortunately, the annotation slides, dang it. But yeah, way over here, we've got this A, which is shared by, draw, figure this out eventually, by the blue, humpback, fin, and gray whales, but none of the others. And here we've got a T that's shared by blue, humpback, gray, and fin whales, but none of the others. And let's see. Okay. This is hard to do by eye, which is why I'm only giving you a small snippet of data. But we've got 3 characters out of 34 that are consistent with the idea that all whales share a common ancestor that they did not share with the hippo. They're consistent with the scenario of this. They're consistent with the scenario that all whales share a common ancestor that they did not share with the hippo, because there's 3 different mutations that can be explained most simply as happening on a branch leading to all whales, but not to the hippo. Am I making sense there? Sort of. Mildly. Some of you are looking at me like I've just bitten the head off a weasel. Okay. Would it help if I brought in some weasels and what's that? It would enhance your learning experience if I bit the heads off a live weasel. What do you think I am, Ozzy Osbourne? I don't know. I could probably say, hey, you know, over the mountain, right between the ups and downs. It's okay. I got his range. Not sure I've got his vocal quality. Anyway, all right. Okay, so we've got 3 characters that we were able to find that are consistent with a tree that looks like this. And we have 2 more characters that are consistent with a tree that looks like this, because we found 2 characters, I think, that are present in all the whales except the orca, and they're not present in the hippo. So we've got 2 characters that are consistent with this tree, with 2 mutations happening on the line leading to other whales, but not to the orca and not to the hippo. And then we have I think we found three more characters that are shared by all other whales except the orca and the sperm whale. So we have data that's consistent with this being the scenario, with the common ancestor of everybody branching off into a line leading to the hippo and a line leading to all whales. And then the descendants of that common ancestor branching into a line going to the orca and a line going to everybody else. And then the descendants of that ancestor branching into a line leading to the sperm whale and a line leading to all other whales, and so on. And we can come up with a tree that's not guaranteed to be right, because this is science and nothing ever is, but that explains as much of the data as possible. We can basically say, okay, we don't absolutely know what happened, we weren't there, but the largest possible fraction of our data is consistent with this as an evolutionary scenario for how we got whales. Incidentally, we have various ways of testing this, and one of them is the fossil record. There's a number of very early whales that have turned up. Some of the earliest have been found in Pakistan that still have 4 legs. And if you saw one, might well have looked more like a seal than a whale. But we can tell from details of the inner ear that they did spend a lot of time diving in the water. They had an ear that was adapted to withstand long-term immersion under high water pressure. There's one called Pakicetus, one called Indocetus. There's a couple of others that have turned up, like Dorodon. And they still have 4 limbs, and on the basis of their skeletons, we can link them to the lineage leading to hoofed mammals, including hippos, which are an early branch of the hoofed mammal lineage. I mean, even though they've got multiple toes and not single hooves the way a cow does, but yeah, the hippos are actually relatively close to pigs. Okay, with me there. So we can cross-check this against other sources of data sometimes. In this case, the fossil record is really good. It's funny, some of the earliest finds before they made the ones in Pakistan were these giant toothed whales that still had 4 legs. There's one called Zygorrhiza and one called Basilosaurus, because the discoverer thought it was a dinosaur, but it's not. And that's actually the state fossil of Mississippi, and the state fossil of Alabama is the giant toothed whale with 4 legs, which I always think is awesome that they've picked such a beautiful case of an evolutionary missing link in Mississippi and Arkansas. Not Arkansas. Arkansas does not have a state fossil, although I would nominate my cousin, but I digress. Anyway, so we can take data and try to work out a pattern, a branching pattern of evolution that is maximally consistent with our data. And that's basically how cladistics works. There are other possibilities, but this one explains a larger fraction of the data than any other could. And demonstrating that on paper takes a stupidly long length of time, but computers can crunch through this data pretty easily. And what I'm going to have you guys do is go play with some data. And after I'm done talking here, I'm actually going to turn you loose, let you go early, and give you something to do that I need to have in my inbox by this coming Saturday. Okie doke? All right. And again, the computers are going to do most of the heavy lifting for you. So let me get rid of, let's go back to this and shrink this down into a readable size. There we are. And here's what I've done for you. I've already talked about plate tectonics. That should be fresh in your mind. So we already know, based on quite a lot of lines of evidence, including some I just didn't have time to get into. You know, you could teach a whole course in this. But we already have it extremely well established that roughly 200 million years ago, the Earth's continents were glommed together in a big continent called Pangaea. And about 200 million years ago is when they started breaking up. By the way, we know less, we know a lot, but it's not quite as much about where the continents were before Pangaea formed. And it looks like that about 750 million years ago, there was another giant supercontinent called Rodinia. And that continent broke into pieces. And the problem is that because the Earth is round, and I don't care who objects, it's round, damn it, round. If the continents are in one and they break up and start drifting apart, the Earth being round, there's only so far apart they can go before they start coming back together. You know, if you have continents moving on the surface of a finite globe, eventually they're going to end up crashing back together. And it's thought that there's a cycle of supercontinent formation and breakup called the Wilson cycle that takes very roughly 500 odd million years. So at the time when the oldest rocks on the surface in Arkansas were being laid down, the continents were pretty widely separated. By the time the Ouachita Mountains form as the core of North America crashes into South America and crumples up the Ouachitas into a mountain range that might have looked like the Himalayas do today. They've eroded quite a bit since then, but yeah, think of it, we used to have Himalayan mountains in Arkansas. Used to be an even greater challenge getting to Mina than it is today. Anybody ever been to Mina? I don't think you're missing much. Kind of the cultural capital of the Western Ouachita Mountains, which is admittedly not a high bar to clear. Anyway, where was I? So yeah, so Pangaea breaks up about 200 odd million years ago and splits into a north half and a south half, with a big belt of ocean separating the north and the south that's called the Tethys Sea, after a sea nymph in Greek mythology. What's left of Tethys is now basically the tropical Atlantic, the Mediterranean, and the Caribbean Sea and the Gulf of Mexico. Anyway, so we get Laurasia in the north, which is Europe plus North America plus most of Asia, and Gondwana in the south, which is South America plus Africa plus Antarctica plus Australia plus India, plus a few bits and bobs like New Zealand and Madagascar and New Guinea and things like that. I used to have a t-shirt that said, "Reunite Gondwana." You know, I kind of felt like that was a political aim I could definitely get behind. You know, if we just put enough outboard motors on the continents, I'm sure we could push Gondwana back together. Anyway, I digress. And I showed you a number of groups of living organisms that live on some and occasionally all of the continents that used to make up Gondwana, and some that live on the continents that used to make up Laurasia. So Laurasia has dogwoods and oaks and bears. Gondwana has galaxiid mud minnows and funny earthworms and lungfish and southern beaches, and in particular a group of conifers called araucarias. Araucarias include Norfolk Island pines. They include a South American tree called the monkey puzzle tree. I was just looking up one of the species that I've got some data on is an endangered Brazilian tree called I will not pronounce this correctly, but something like the Pinheira do Paraná. Pinheira do Paraná. It's not actually, I mean, Pinheira is a pine tree, right? It's technically not a pine. It's not a North American type pine, but it is a conifer. And it belongs to this family called the Araucariaceae. And apparently it's critically endangered, but that doesn't stop the seeds from being considered a delicacy in, especially way down in the south in the province that I'm not going to pronounce correctly, but it's something like Rio Grande do Sul. Do Sul? Do Sul. Okay. Way down in almost Uruguay. Okay, right. So what I've done is drawn up a whole bunch of data, and this is publicly accessible at a website called the National Center for Biotechnology Information. It's part of the National Institutes of Health. Basically, if you do any federally funded science and it generates DNA sequences, you have to upload the sequences to this giant database that's been in existence since the 80s. and has an absolutely ridiculous stomping amounts of DNA sequence data in it. Complete genomes of now well over 1,000 different species and more added, almost every day. And isolated genes from hundreds of thousands of different species. I mean, it's still a drop in the bucket, but the drop is getting kind of big. And I went there so that you don't have to. And I found this. Let me see if I am. I'm going to file share that there. That is sequences for a gene that's critical in photosynthesis called ribulose bisphosphate carboxylase, aka Rubisco. Any of you guys remember Rubisco? Yeah, it's the enzyme that does a lot of the work in actually grabbing CO2 molecules and sticking them together. Yes? Well, there's the sequence right there for the Rubisco gene for a very rare conifer from Australia called the Wollemi pine, or Wollemia nobilis, which is neat because it was only discovered still alive roughly 20 odd years ago, but it was known as fossils from much farther back. They thought it had died out, but in fact it has not. But it's restricted to only a couple of places in Australia, that the locations of which are being kept secret. Because of course, as soon as we announce the existence of anything precious and rare and beautiful, somebody has to go and try and F it up, because people suck. Anyway, so that's sequence data for the Wollemi pine. And below, that's another Australian tree, Agathis atropurpurea. There's one for a New Zealand tree, and one for Indonesia, another from Indonesia, one from Papua New Guinea, one from some Pacific islands, Australia again, Australia, Pacific Islands. There's one from Chile, Araucaria araucana. I think that's called the monkey puzzle tree. There's another from Australia. There's one from Brazil, Araucaria brasiliensis, Australia again, Papua New Guinea, New Caledonia, okay. And then a couple of conifers from the US and Japan, and one from Taiwan. Okay. Okay, so what's the point of all this? If plate tectonics works out, what we might predict is that all of the conifers on Gondwana would be likely to share a common ancestor with each other. That's not an ancestor of the conifers in North America or Asia, right? Yes? Because presumably after the split, conifers must have diversified on Gondwana, but species of conifer on Gondwana could not easily disperse back to Laurasia because the continents are now separated. So just like the marsupials of Australia or the xenarthrans, the armadillos and sloths of South America, the conifers on Gondwana must have diversified there, which means we might predict that they should all share a common ancestor that is not an ancestor of the conifers on Laurasia. Am I making sense there? The family tree, you would predict, should mirror the breaking up of the continents. And in fact, we can do better. We might predict that all of the Australian trees ought to share a common ancestor, and all of the South American trees should share a common ancestor. Because when Gondwana started breaking up, some of these conifers should have ended up in South America and diversified on their own there, and others would have ended up in Australia and diversified there, right? Am I sort of making sense? We might predict that the branching pattern of the conifers ought to mirror the breakup of the continents. Okay, well, let us see if that actually works. All right, this is okay. The www.phylogeny.fr. Okay, what am I sharing? Okay, hopefully everybody can see that. I think I'm properly sharing this page. Okay, we don't need to know everything that this page is capable of doing. Okay, there we go. It was loading a little funny, but there it is. And what we can do is scroll down. I'm going to click on one click. And I'm going to take the data set that I just showed you guys and upload it to this website. And the website is taking all that data. First of all, it's coming up with ways to line up all of those blocks of DNA, and there's some heavy math involved there. And now it's cranking through a mathematical algorithm to find the tree that fits that data best. And I'm going to dim the lights for just a sec. And it is going crank, 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 boom. Okay, let's see. Okay, look at this. There's our tree. I don't know if I can expand that, but look there, up at the top, we have two species from the United States, one from Japan, one from Taiwan, and then another from Japan. So those are all of the conifers that we included that would be native to Laurasia, right? Yes, see it? So our simplest hypothesis is all of the conifers in Laurasia descend from a common ancestor that's represented by that branch point right there. Now, we might need to refine that because we have not tested all of the conifers in Laurasia. We only tested a few, but for right now, you know, this seems to work. Now, let's look at the rest. We have a whole bunch of genus Agathis that all seem to descend from a common ancestor, and they're all found in Australia, Indonesia, nearby Pacific Islands, Papua New Guinea, and then there, Wallonia, that's Australia as well. And here we've got a bunch of genus Araucaria, and they all come from a common ancestor. And right here, we've got those two Araucarias that are native to South America. So again, we could always refine this by adding more taxa, adding more data. You know, you can always add more data, but it does look like that the Araucarias that we have in South America, the ones that we've got, appear to share a common ancestor. So it looks like this is working reasonably well. The Araucarias existed on Gondwana and diversified while Gondwana was breaking up. So we do have one branch right here that consists of the South American species that got stuck on South America when South America broke away from Gondwana. And then we have a whole bunch that are found all over Australia, New Zealand, Papua New Guinea, which used to be connected to Australia by a land bridge, and various Pacific islands that seem to have evolved in place. Am I making sense? Okay, so you see our cladogram does seem to have this deep split between Laurasia up top and Gondwana down at the bottom. And we could always use more species, but it looks like we do have one branch right here that contains all of the South American species. Okay, isn't that cool? All right, the way you can put together studies of the evolution of a group of species, and sometimes you can fit that together with the evolution of the surface of the entire Earth, the configuration and the movements of the continents. Here is what I'm going to have you clowns do. Okay. We know that when Gondwana started breaking up, the first parts to separate were Africa and India. Now, there's two possibilities. One possibility is that the conifers of Africa and India evolved after the breakup, and they never had much in common with the conifers of Australia and South America. Australia and South America were the last two continents to split. So it's possible that African and Indian conifers, remember, Africa and India have both crashed into Laurasia, right? India is now jammed into the southern side, the south side of Asia, and Africa is slowly drifting north. So much so that there is a land bridge from Africa to Europe and Asia. It goes right through the Middle East until we cut it with the Suez Canal. But yes, it's still there. And there's evidence for some dispersal going on between Europe and Africa. One example I can think of is the fossil they found that so far looks most like a plausible ancestor of gorillas actually turned up in Greece. So great apes may have actually evolved in southern Europe and then dispersed across to Africa, which is where they're found today. There is still room for argument about that, but it is possible that the conifers of Africa are descended from Laurasian species that dispersed as Africa started getting close. And it's possible that conifers in India are also descended from Laurasian species that have dispersed in once India docked with Asia. All right, let me cut over to the screen share, the whiteboard, because I think I can make this maybe slightly less confusing. Okay, there's the world. No, it did not really look like a potato. I'm just drawing an equatorial projection. Let's say about 100 million years ago. North America is kind of over here. Europe is over here, and of course that's Asia. So there's North America and Eurasia. I'll just write EU right there. So that's your Laurentia, or Laurasia right there. Gondwana is South America over here, and Africa over here, and India over there, and then Antarctica is right there, and there's Australia with a few bits and pieces like New Guinea is here, Madagascar is right there, New Zealand is somewhere off here. But that's our basic scenario. We've got an ocean separating the two, and it's called Tethys. Sorry, I can't write that clearly. And then we have the Gondwanan continents down there. Now, we've already seen that Araucarias evolved on Gondwana, Because if we were to take that tree that we got and lay it down on the continents, the conifers have some branches in North America and Europe and Asia, and then another bunch of branches that are found only in Australia and New Zealand and Papua New Guinea and some of these islands and things, and some that are found in South America. So we could take that tree that we've got and almost lay it down on top of the map. Yes? Here's what we want to know. One possibility, it's a question mark, is that conifers in Africa and India are most closely related to the conifers from Gondwana. Yes? Right? It is also possible, however, and here's a second possibility, and I'll do it in a different color, that conifers of Africa and India are most closely related to Laurasian conifers, and they dispersed into Africa and India when Africa and India docked with Laurasia. You know, when Africa got close enough to Eurasia to create the Middle East, which is basically the land bridge between them, and when India got close enough to start pushing up the Himalayas. So are the conifers of Africa and India Laurasian species that have dispersed, or are they Gondwanan species that have vicariated? Have there been conifers in Africa and India from Gondwanan days, or did they disperse there at a much later date? So we've got two scenarios, and off the bat, we don't know which one is true. Okay, with me there? All right. Would you like a couple of minutes to ponder that? Would you like a couple of minutes to go get some coffee or something? Sorry, I'm noticing a couple of people that I'm clearly not thrilling them with my sparkling wit this morning. But I tell you what, it is 10:10. Take about a 10-minute break, and when we come back, I'll show you how to try to solve this, how to figure out which one of these scenarios is likeliest to be correct.